Okay, thank you, SJC team, for this opportunity to come back here and present on a much different topic than what I presented on in the fall. So as everybody knows, our topic for today is how to read a scientific article. And let me first state that it's not easy, as a lot of you know, and it's not meant to be easy, but it gets easier in time. So a lot of you, the first time you read an article, you may feel like you're looking at algebraic formulas, calculus formulas, or reading a foreign language. And that's okay. And that's partially normal in the beginning. So my first question is, what is a scientific article? And this is from a student's perspective. Students may think it's an assigned reading by professors to make them feel like they're out of place and like they don't belong in their scientific club. I assure you that's not what we're trying to do here. Another student may think that it's a collection of scientific findings that appear to be written in a weird, strange, nonsensical, peculiar, foreign scientific language. And that's not exactly true either. So what exactly is a scientific manuscript? Why do we care about these? Well, scientific manuscript from a basic standpoint is it's a publication based on empirical evidence, right? So seems like a pretty simple statement, right? Do you guys know what the word empirical means, right? I imagine there might be a few of you who aren't exactly sure what that means. And this is really the essence of reading a scientific manuscript. If you don't understand something, you need to look it up. So empirical means, by definition, it's originating in or based on observation or experience. So when we talk about publishing data based on empirical evidence, what we're really talking about is, going into a lab and doing wet lab work or doing computational work, collecting data, observing that data, and then sort of reporting on it. But that's empirical evidence. So it's based in observation. And my first tip is this: do not be discouraged or feel inferior when reading a manuscript. And I know I certainly did in my beginning stages of a scientist. So don't do that. Just accept it in the beginning that it's going to be a little tough, but you will get through it. And I can assure you, you will get better in time. Now imagine we probably have a bunch of maybe 18 to 22, 23-year-old people here. And if I were to give you a scientific manuscript, even though you feel like you probably couldn't read it in its entirety and fully comprehend it, if I were to give it to you, and if I was to give it to this person over here on the right side of our screen, who's, I don't know, 2 years old or something, maybe a little less than that, who do you think would read it better? Obviously you, right? Well, it turns out that this young toddler, this individual here, grew up to be a very famous scientist who doesn't always take himself so seriously, right? Albert Einstein has his tongue out right now. And there was a point in time when Albert Einstein and other very prominent scientists in various fields, they had to read their first scientific manuscript. So in Einstein's case, was probably about 3 years old. But he did struggle the first time he read a scientific manuscript. We all did. All the professors that you have in all of your STM classes, we all struggle. But you get better in time. So if you accept that and you recognize that, and if you don't feel like it's your fault, or if you're not intelligent enough or you don't belong, just get those thoughts out of your mind initially and right away. So let's discuss what the origin of a manuscript is. And this is really just my interpretation. Today's presentation is all my interpretations and based on experience. So you come up with a creative idea, and that creative idea could come from someone like yourself, could come from a professor like me, or maybe a very seasoned professor who's been in the field for a very long time. Creative ideas can come from anyone. So never assume that it's only going to be someone much older than you. It can come from you. And you take the creative idea, and what we do is we write a grant. So we go through the stages of grant writing, and what we're trying to do is secure funding. And what funding is, we obtain money from different services or different organizations, such as the National Institutes of Health, the National Science Foundation, even philanthropists like Bill and Melinda Gates. They donate a lot of their money for scientific research. So as the kids would say, you trade this idea to secure the bag. And with that bag comes a lot of money. You could hire people to work in a lab, and you can conduct the research. So the idea is you are going to try to realize those original ideas and those hypotheses that are written in a grant through experimentation, right? Through empirical type of evidence. And if you are lucky enough, you will come across some sort of scientific discovery, and maybe a big one, right? Such as the discovery of DNA. Let's go back into the 1950s, right? And that was the age of DNA. We learned that DNA is the genetic code. So it stores a lot of really important information for our cells, for our proteins. It's a double helix. We learned the structure of it through Watson and Crick, but more importantly, Rosalind Franklin. So we learned so much, right? And if you learn these things, you need to publish the data. You need to take that data, report it to the scientific community, but in addition to that, the general public. If someone, right, my mom or my dad, they could spend money, I don't know why they would, but spend money on the scientific journal Nature. And they may have a subscription to Nature and read that on, every 2 weeks or every month, whenever it's being published, right? So the information that we publish, it's really open to everybody, as long as you have access to it or you could pay for it. So I want to, sort of, before we go into how to diagnose a scientific manuscript, let's look at it from a slightly different perspective. And we'll talk about that now. So in order to understand how to read one, let's talk about the thought process of actually writing one. So you can see it from a different perspective. And the basic parts are an introduction, methods, results, figures, discussion, and conclusions. And all of you are familiar with these. So this is the thought process, and this is what I came up with. And again, please remember that this is my opinion. I'm going to provide additional resources for you that you can have a look at too. And you might find there's something else that is better or is more in line with your thought process. Or from my experience in interacting with colleagues over the years, this is generally what happens. So we start in the beginning. We create figures and tables based on that empirical evidence, and we assess it. We look for statistical significance. And if we have a finding that looks good, it's sort of like jumping out of this on the paper, then we will say, all right, it's time to write a science manuscript. So you put together the figures and the tables, and then we move into the results section. For the results section, we essentially just describe what we see in our figures and our tables. And routinely in a results section, you're going to see references to those figures and tables too. Now, some people, they may write a materials and methods section after the results section. Others will, that might be the first thing they do. Maybe they just aren't in the mood to write a scientific manuscript and they need to ease into it. Generally, writing the materials and methods section is probably one of the easiest parts. So it's easy just to, let's just get started, and I'm going to do some mindless writing right now. And as soon as you get started, you feel better and it gives you more inspiration. So again, some people will do a materials and methods section after the results. And then something that makes sense is we go to the introduction. And the introduction section is the background, its rationale, you're describing your hypothesis, you're sort of setting the stage for what the reader is going to learn about a little bit later as they progress through the manuscript. Then I have this phrase, and it's shown in red. And the key on the bottom, it says, valuable parts of a manuscript to extract information when reading. And that's sort of my pitch today. There are certain regions in the manuscript where you can find interesting nuggets. And the last paragraph in the introduction is generally one of them, as it has information about the experiments that were performed, has a synopsis that's important. So that's an area that we're going to focus on a little bit later today. Following the introduction, one would write the discussion section, and that's where you report your significance of the findings to the field. You're going to compare and contrast it to other reports. You're really discussing what you observed in detail, and you're providing substance behind that. And the first paragraph of the discussion, in some cases, it looks like the last paragraph of the introduction. So it's going to have, again, a recap of what these findings are. The last paragraph of the discussion, that's conclusions, applications, future experiments. Those are all the lofty goals, right? That's where I write my manuscript and I say, whatever I did, I don't even care what I did, but I'm going to cure cancer, or I'm going to send somebody to Mars, right? That's where these big hopes and dreams, probably with 95% of manuscripts, it actually never comes true, but that's what we do in the last paragraph. So it's going to give you sort of a broader horizon look on what these researchers actually think of their own self-worth as a scientist, but also the worth of their data. And lastly, abstract. And abstracts, they provide overviews of manuscript. And I think most of us, we know that. So I thought the best way to really approach this topic, and to be honest with you, I think this could be like a 5-part series. And we could come back 5 weeks in a row, keep working through this. It's almost impossible to learn how to fully break down a manuscript. And I don't know if anybody knows, right? There's not one easy way. You have to just read it. You have to look up information that you don't understand and keep progressing. But we're going to provide you with a few tips. So as Nanditi mentioned in the beginning of in the introduction today, we're going to use a manuscript that I spoke about in the fall 2020, and it's titled Alpha Defensin 5 Differentially Modulates Adenovirus Vaccine Vectors from Different Serotypes in Vivo. Now, I'm going to put myself into 20-year-old Larry, and I would have no idea what that means. The only thing I might know is modulate, different, and in vivo, but I'm not sure how good my explanation would have been when I was 20 years old about what in vivo really means. So one of the best things, and we're going to use a title as our sort of our approach here. We're going to interpret the title of the manuscript, and that's tip 2. So what I've done here was I've looked at this title and I broke it down. I said, all right, what is an alpha defensin? Because I had no idea. What is an adenovirus vaccine vector? I don't know. What is a serotype? And what does in vivo mean? Those are the questions that I'm asking. And we're going to see how we can pull together information to help us, fill this narrative and look, this isn't exactly black and white, right? We're going to look for these open gaps that's actually in this title and provide more substance and more clarity to it. Because, when you're 20 years old, you're sort of a novice in the field. So it's going to take some time to learn. So the first question is, what is an alpha defensin? And my suggestion is to briefly skim the abstract or the introduction to get this answer. So looking through the abstract, it says alpha defensins are effector molecules of the innate immune response. All right, I'm not exactly sure what an effector molecule is, but I've heard of immune response before. So we can assume it has some sort of role in immunity. And the second part of it says it has been shown to modulate natural infection with adenoviruses. All right, so an alpha defensin has some sort of effect on adenoviruses. So the best thing you could do is start summarizing some of this content, whether you want to write it down, you just want to verbally say it to yourself. Alpha defensins are part of the immune system and have a role in affecting adenoviruses. That's provided a little bit more context. I took it a step further and I went into the introduction, just really into the first and second paragraph. And what you see in the introduction, especially if you look in the first paragraph, all right, my little cursor here, alpha defensins, they're small. They're these cationic peptides. They're 18 to 45 amino acids. They have 3 disulfide bonds. It's a lot of general information about what an alpha defensin is. But remember, I'm sort of focused on what an alpha defensin is in the context of an adenovirus vaccine vector. So I keep reading. And then the second paragraph reveals some of the information that I'm looking for. The first line, alpha defensins, they exhibit potent antiviral activity against adenoviruses. All right, so now we're talking, right? Now this is starting to provide a little bit of substance on what that title actually says. In vitro experimentation shows that defensins, they inhibit adenoviruses from specific species, and alpha defensins, they can also augment, which means enhance, adenovirus expression from other species. So I don't exactly know what species are just yet, but what I have seen here is that defensins, they have a distinct role, 2 distinct roles. One, they can enhance adenoviral infection. The other is they can inhibit it. So I summarized that again in my own words. So I said alpha defensins, they have antiviral activity and can inhibit and enhance an adenoviral infection depending on the specific species. And because I didn't know what that meant, I thought, well, maybe it's like a class or a specific type. So I put these other words in there too to help me understand. So what we can do now is we can take these 2 statements that we came up with, right? And this literally took 30 seconds, maybe 1 minute to find this information. And we're going to combine it. And we're going to call this our iteration 2 of our working summary. And we're going to state alpha defensins, they're innate immune system molecules that can enhance and inhibit adenoviral infection, depending on the type. So that's our working summary of what an alpha defensin is. And right now, that's probably good enough information that I could proceed with this manuscript and have a good idea of what's going on. So the next question we have, what is an adenoviral vaccine vector? So instead of looking in the manuscript, I want to show you guys the different approaches that you can take. The second approach is we're going to take advantage of Lehigh's awesome resources. We're also lucky that Lehigh spends a lot of money on scientific journal subscriptions on an annual basis. So we really, the world is our oyster as far as scientific literature is concerned through Lehigh University. And what I'm going to show you here is, I want you to put yourself in a perspective. You're taking a class and your professor assigns you and 3 of your classmates this article to read. And your specific job is to learn about adenovirus vaccine vectors, right? It's not about alpha defensins or what in vivo means. It's specifically about adenovirus vaccine vectors. One thing that you can do, you could take advantage of the power of a review article or a review manuscript. And what that essentially does, it's not based on, you aren't presenting and publishing original data. What you're doing is you're stopping and you're assessing what the current climate of, adenovirus vaccines look like as of today, right? So let's just say you were to write a review manuscript on coronavirus. Things you would include in that, the life cycle of coronavirus, right? Infection rates. You'll talk about the Moderna, the Pfizer, the Johnson & Johnson vaccine, right? It's going to be an all-encompassing overview of that specific topic. So there's a way that you can go about doing that. And I'm using adenoviral vaccine vectors as an example. So I've created this video, and right now it's showing me going to the Lehigh University library page. And what I'm doing is I'm going to the database section, and you're going to see that Lehigh has subscribed to lots of different databases. I suggest you just peruse and you go through this list. But what we're really focused on is something called PubMed, which is essentially a database where scientific literature is deposited and we can access it through PubMed. So that's what I'm doing right now. A lot of you probably know about it, but some of you may not. And once I get there, I'm going to bias my search. So I'm typing in adenovirus vaccine, and I type in review because I'm looking for a review manuscript. And you're going to see a couple of cases here where I use the little cursor, and right there, I'm showing that means these manuscripts are review manuscripts. So I'm perusing through and I'm looking through and trying to identify which one makes sense. And it says adenoviruses as vaccine vectors. So I click on that. And as I mentioned, we have a lot of free access to certain journals. Now you have access to a wide range of topics that fall within adenoviruses as vaccine vectors. There's a standard abstract. There's going to be an introduction that's associated with this manuscript. You can learn about the adenovirus genes and the genomic architecture, which you'll see right here. You'll learn about adenoviral infections, different types of tropisms, meaning what cells do they infect. You'll learn about immune responses against adenoviruses and actually how to construct them and to actually make a vaccine vector, and a lot of other important information. So you can become an adenoviral vaccine vector just by going to this review article. So if you feel like an original piece of data that you are studying, it doesn't make sense or you need more context behind it, you should use a review article. It could provide so much more substance for you. So what I did in this case was I just went into the introduction. I looked for the first kind of few lines that made any mention about what an adenovirus vaccine is, and I'll read it to you. So it says, while adenoviral vaccines, their reduced appeal as a gene replacement vehicle, it invited their use as a vaccine carrier. Adenoviral vectors are attractive candidates for transfer of foreign genes for a number of reasons. So my interpretation of that is adenoviruses act like a vehicle delivering foreign genes for vaccine purposes, right? Just a very basic, simple summary. Now I have a better idea of what an adenoviral vaccine is. And if you are more of a visual person, feel free to create images, right? You could almost create a story dialogue through images. So whatever your modality of learning is and understanding, I say use it. All right, so now we've answered a couple of questions. Now we're going to go to what is a serotype? And we're going to jump onto Google in this case. And I search for antivirus serotypes. Just clicked on the very first link that popped up. And I'm going to bias my search very quickly by doing Control F and searching for the word serotype. And I want to see if there's a description there that's going to make me feel confident that I could explain what a serotype is to somebody else if I had to give a presentation on a literature review piece. And it turns out, I think the third time I searched the word, there is a paragraph here, and it's actually within the abstract, that discusses what an antivirus serotype is. So we're narrowly focusing in on this. There are 51 recognized serotypes of antiviruses, 85 genotypes based on their unique genomic characteristics. So as soon as I see genomic characteristics, I'll automatically think nucleic acids. And then it goes on to say, antivirus types are classified into 7 species. That's where that word species pops up again. Based on their, excuse me, based on their analysis of their genomic DNA sequences. And we know what DNA is: pathobiology, an immunology word, and then tumorigenic properties. So because you don't have to become an expert on this, you just want to have a general idea of what a serotype is, I just narrow it down to a couple of things that make sense to me. So I don't exactly know what tumorigenic properties are, right? So I do know, but I'm putting myself in the mind of a 20-year-old. And pathobiology, that might sound a little confusing, but you do know what DNA sequences are, and you have heard of the immune system before. So we can say that antivirus serotypes are grouped based on DNA sequences and properties related to the immune system. And that's enough information for us to understand what a serotype is. And then the last question that we're asking at this point is, what does in vivo mean? And in vivo refers to when research or work is done within an entire living organism. Could be an animal model or a human clinical trial. So then they actually compare and contrast with the word in vitro. So now I have a better understanding, and I probably knew what in vivo meant, but I might not have been able to explain it to somebody. So at this point, we've sort of answered all these questions that I had, and we could take these brief summaries, which really, in essence, is no longer than just a few minutes of some quick searches. And we come up with a new iteration for what that title meant. And now I'm stating alpha defensins enhance and inhibit antivirus vaccine delivery vehicles, which are classified based on sequence and or immune properties. The study was conducted in an animal model system. And for me, the novice scientist would understand this a lot more than this. So now I feel a lot more comfortable going into the manuscript, having a general idea of what may have been done. And the beauty of this is you'll see how accurate you were, or maybe you weren't. So tip 2 summary is use different resources to look up information. I can't tell you how critical, how important it is. There's no real magic bullet on how to decipher scientific literature. It's not easy. It's not inherently easy, but it does get easier with time. And you'll see examples of that. So take advantage of other resources. You have PubMed, you have access to many different journals. You could look at original manuscripts, review manuscripts. These are all peer-reviewed manuscripts that are published. They've been reviewed by experts in the field. So the literature, the publication should be relatively strong. You could also do simple Google searches, right? I promise you, not every single professor who's looking up terminology for a manuscript is going to go to PubMed. There are times when we might use Wikipedia just looking for a very quick answer. Now, I'll tell you right now, Wikipedia could have, in a lot of ways, Wikipedia could be pseudoscience. You don't really know who's on the other end entering in that information. And I have seen, drastically obscene comments in Wikipedia regarding scientific fact. But, in general, you might be able to get an answer there, or, whatever those first few links are, they pop up on Google, you get a quick answer. So depending on the question that you're asking, how important the accuracy is of it, you could do a quick search. So the take home point is, when you don't understand terminology, you need to look it up. And I can't stress that enough. It's probably the most important thing I could tell you today. My tip 3, Understand that reading a manuscript is sort of like looking at an hourglass. Here's an hourglass. And I like to call this hourglass trends of a manuscript. And what this really means is we don't care about what's inside the hourglass, if there's a little sand particles or whatever that may be, right? What we're really focused on is the shape of the hourglass. And it sort of starts wide and broad, and then it goes in, gets more narrow, and then it broadens out again. So we can group this if we compare this to scientific literature, where the top portion, these are general type information. We have a very specific information, and then it sort of gets general again. So by definition, and with an example, general is a general knowledge related to the field, right? So if we go back to alpha defensins, their immune system molecules, their cations are about 30 amino acids in length. They have antibacterial, antifungal, antiviral activity. I didn't talk anything here about an adenovirus vaccine or the real crux of this manuscript, right? This is sort of what came from that first paragraph in the introduction, which I mentioned before, right? So this type of information would obviously start up here in that more general section, whereas a specific section is something that's directly related to your findings or your findings themselves. And that is alpha defensins. They inhibit some adenoviruses, and they also enhance other adenoviruses. And that's data that was, empirically determined, and it came from this manuscript. So let's put this in a little bit more context. The beginning of an introduction, at least probably the way 75% of manuscripts are written, the beginning of an introduction is going to be more general information. The middle of the introduction gets a little bit more narrowly focused. And then by the end, the last paragraph of the introduction, it's extremely focused. The materials and methods, they are embedded in, the core of your data, which is, all original research. So of course, that's going to be specific to what you're doing. The results and figures, that is the crux of the manuscript. That's very specific. Then you go to the beginning and the middle of the discussion, and in a lot of ways it's specific, but you're starting to make comparisons to the field in general and look at other scientific research. And then by the time you get to the end, right, you're literally saving the world, and that's it couldn't be any more general at that point. So that's my classic breakdown using an hourglass approach. And I challenge you, the next manuscript you read, just pull this image up, and I have like this little rubric at the end that you guys can see or take with you, and then sort of break it down, right? It's easy to get overwhelmed when you read a manuscript because there's so much terminology that you may not understand. There's just so much information, but there's certain keys in that manuscript where there are nuggets that will help you better understand it. And we're gonna talk about some of those. So now that we broke down the title, we have a running summary. Let's see what else we can tease apart from specific components of the manuscript directly. And this is where these nuggets come into place. So I mentioned this last paragraph of an introduction. I'm not going to read this directly, but what I'm going to do is I'm going to show it again with some of my annotations. And I recommend annotate the heck out of your manuscripts that you're reading, whether you're doing it online or you print it out and you read it. You should have a manuscript that is going to almost looks like there's another manuscript written within it, because you have annotated so much to try to understand it. So the first part of the last part of the introduction is telling us what the authors did, but it didn't reveal any of the results. So basically what we state is there's certain adenovirus serotypes that were assessed in different ways. And there's also a mention of AD5 and AD26, specifically vaccine vectors, right? But it didn't reveal any of the results or findings. It's the second part of this last paragraph that does. And what it basically says is the latter part reveals that adenovirus 5 is inhibited as a vaccine vector by defensin, while AD26 as a vaccine vector is enhanced by defensin. And there's also other terminology in here, such as innate cytokines, antibodies, CD8 T cell responses. Just keep in mind, the first time you skim through a manuscript, you don't have to look in excruciating detail, right? You could leave out some of this information. If you just group this into immune responses, that might be enough for you. So we take that information, and now we can add it to our running summary of what we had going on. So our third iteration was alpha defensins, they enhance and inhibit adenovirus vaccine delivery vehicles, which are classified based on sequence and or immune properties. The study was conducted in an animal model system. So what we've added from just looking at the last paragraph of the introduction is that we're specifically talking about AD26 and AD5. So instead of just this general adenoviruses, now we're a little bit more specific, and we're sort of gaining ground on what these authors did. The first paragraph of the discussion, and this has been mentioned other places too, but I want to point out, we mentioned the word mice, and these authors, they were looking at the protective efficacy of AD5 and AD26. Little caveat, this AD26 vector, this exact one is the one that was just approved by Johnson & Johnson. It's what's delivering the spike gene to humans, which is awesome. But in addition to that, so we mentioned the word mice, which has to do with the model system. So instead of just saying this animal model system here, now we've added mice to that. So our working summary is alpha defensins, they enhance AD26, inhibit AD5 vaccine immune properties in mice. The last paragraph of the introduction or the discussion, and this is where I mentioned about some of these lofty expectations. I'm just going to highlight a small portion. So the authors state that such knowledge might facilitate the idea of using alpha defensin therapeutically to inhibit ad type sensitive to HD5, like AD5, but also can be explored for adjuvant discovery with ad type similar to AD26. So that information we essentially just tacked on here. And all we really stated is, in my own words, this data is potentially significant because to me, it sounds like we could use a defensin to inhibit some adenoviruses, and we didn't go into this, but adenoviruses, they cause the common cold. So it may be used as a drug to ward off certain adenoviruses, but it could also be used as a way to adjuvant or enhance the vaccine immune properties of serotypes like AD26. So when we compare our first iteration to our last iteration, we have a pretty good understanding at this point. And, you know, it may seem like this takes a lot of time, but the truth is it doesn't. We looked at the title, we broke the title down. Within 5 minutes, you could get explanations what each one of those words mean, and probably more people than I would expect. You guys probably already know what a lot of that information means. And then we went to the little bit of the introduction, the last paragraph of the introduction, and the first and last paragraphs of discussion. That's it, nothing else. And we have a working understanding of what these researchers did in this manuscript. So if you could do that, and then you move into the figures and the crux and the core of the manuscript, so the figures, the tables, and the results, you're going to have a pretty good understanding of what's happening. So I like this approach, and this approach has worked for me in the past. So one thing I want to note here, notice that we have not examined the abstract in detail. So you may be saying, Larry, why are you wasting our time? We could just look at the abstract. And that's true. And probably 95% of the time, you're 100% accurate. So in general, that's one of the first places where you want to look, right? However, on occasion, sometimes abstracts can be biased, as the author is only highlighting or boasting about the good things that were reported on, right? I like to look at it like this. Have you ever seen this quote from Chris Watt? When you meet somebody for the first time, you're not meeting them, you're meeting their representative, right? So you go on a date with someone, right? You aren't going to show them how crazy you are. You're going to show them the best version of you. And that's how an abstract is written in a lot of ways, right? It's going to be biased. Scientists like myself, researchers, we understand, we recognize that when people are looking at manuscripts, they're going to read the title and the abstract. So you want to put your best foot forward with the abstract, which means maybe you're biasing things a little bit. And as a reader, you may be biased by reading the abstract because it's going to be written in a certain way that's really trying to sell a certain idea. And I challenge you not to read the abstract. I challenge you, if you're ever put in a situation where you have to, present an article or such, don't read the abstract, right? Read that last. Come up with your own abstract as you progress through these different stages in the manuscript and see how accurate you are, right? Your interpretation of the data may be a little bit different than those authors' interpretation of the data as they state it in the abstract. So that's why I said that. And that's why we haven't really focused on the abstract. And I also wanted to take, does anybody have any questions at the moment? Okay, so we'll keep moving. So now we're going to talk about figures. And this is the first figure in this manuscript. And we aren't even going to look at the data yet. We're just going to read the figure legend and see if this makes, sense to us. And the title of this figure is Analysis of Adenovirus Sensitivity to HD5, which HD5 is alpha defensin 5. I didn't really say that, but it's not extremely important now, in A549 cells. So I feel comfortable that we have a good understanding what an adenovirus is. We generally know what alpha defensin is, and we know it's going to inhibit and enhance certain adenoviruses. I have no idea what A549 cells are. So we could let's just ignore that for a second. Then it mentions 4 different adenovirus serotypes that were incubated with, looks like different concentrations of defensin from 0.2 micromolar to 50 micromolar alpha defensin 5, or 50 micromolar of mutant HD5. So I have no idea what mutant HD5 means. There's also another term over here. So these adenoviruses are expressing something called EGFP. Let's just assume we don't know what that means. So again, we go back to what we did before. Let's try to identify, to fully understand this manuscript, what this information is telling us. So we're asking the question, what are A549 cells? Asking another question here, are these physiological concentrations of alpha defensin? And we aren't exactly going to answer this today, but that's a question that you should ask yourself. What's a mutant HD5? And what does this acronym of EGFP stand for? So let's head to the results section, right? Remember that figures, they are tied to the results. So there might be more information there. Let's see if we can find it. And after reading and just biasing my search to go to the first figure, I come across a lot of information that was pretty helpful in answering my questions. So number one, immediately I see, it says something about these adenoviruses, they're expressing enhanced green fluorescent protein. So that's called EGFP. That's what EGFP is. And some of you may have even worked with that in a biology lab, whether in high school or here at Lehigh University. Okay, so now we could understand what that acronym stands for. The second point, we had no idea what A549 cells, and it just so happens that the authors explained it. We selected A549 cells for initial tests with HD5, as A549 cells are often used to study adinfectivity. So in my opinion, in my own little summary, A549 cells are a standard line that are used for adinfectivity studies. And there's even some references here too. So you could feel free to go back to these references if you want to see how other researchers conducted different experiments using ad vectors and A549 cells. And then one of the other questions that we had is what is this mutant HD5? So there's a comment here, it's a negative control mutant HD5. In my opinion, I don't think that's enough information for me because I don't know what a negative control is. Like I've heard of negative controls, but I don't truly know what one is. So let's examine it in a little bit more detail. So if it's not shown, if it's not described in the results section, then it might be an actual experimental setup, which would be in a methodology. So we're going to go back to the methodology now, which we haven't looked at because remember, you don't really care about the methodology unless you really need to know it for a reason. And all I do is I just do a control finds and I look for the word mutant. Now I found mutant, it says mutant HD5. Perfect, that must be sort of the information I'm looking for. Over here it says synthetic HD5, which must be the normal one. And it has some sequence information as well, right? This is amino acid sequence, and there's another one associated with the mutant. So if you do a quick comparison, what you're gonna find is there are different amino acids that have been substituted here compared to this region. So that must explain what mutant is, but I'm still confused because I don't really know what a negative control is. So I have to ask myself, what is a negative control? And I just come to Google real quick and I type in, what is a negative control in an experiment? And quickly, Google tells me negative controls, there are particular samples, including the experiment, that are treated the same as all other samples, but are not expected to change. Oh, that must be why they're negative. And then, these are the type of things sometimes when we can just relate it to real life. I know a lot of you have been tested for COVID recently. If you're positive for COVID, it means you have COVID. If you're negative for COVID, it means there's been no change, right? You don't have it. And that more or less you could relate, real biology examples in the life around you to, scientific literature and experimentation. And in my opinion, the moral of this story again, I'm going to keep repeating, it's not, there's no one easy way to do all this, but I'm going to show you the beauty of just immersing yourself and asking questions. What we've done so far is we are adding to our biology toolkit. We've learned about mutant and negative control, so we have a better idea what those are. We learned what that acronym stands for. It's enhanced green fluorescent protein. We know what a serotype is, at least in the context of adenovirus. We know A549 cells it's a permissible cell line for adenoviruses. Alpha defensins, we have a pretty good understanding what defensins are. We know a lot about vaccine vectors with adenoviruses because we looked at a review manuscript. And then we've also had a simple definition of what in vivo means. So what this is going to do for you, and this is really important, the next manuscript you read, or maybe the next 5 manuscripts you read, you are going to keep building your biology toolkit. So let's just say 5 manuscripts from now, you come across another one based on antivirus vaccines or some other vaccine. It doesn't make a difference. And they talk about mutations, or they talk about a negative control in an experiment. You don't need to go back and look up what a negative control is, right? You don't need to look up what a serotype is, or you don't need to understand why they use A549 cells, right? You already have that information as part of your biology toolkit and your biology repertoire, and you take that with you. So every manuscript you read, you keep feeding that scientific beast that is growing in your mind. And as time passes, scientific manuscripts, they are easier and easier and easier to understand. And I'm sure most professors can tell you that. Tip 4, annotate the manuscript as much as you'd like. Still, when I read manuscripts, especially if I'm doing some sort of a lit review and I have to present it, I will write so much information down on that manuscript. Like I said, it's almost like I've written a manuscript within a manuscript. This is just an example. We could go back to that figure 1. And what I'm showing here is I've highlighted something. I have this box around mutant HD5. It says the diagonal box is over here. That's going to be where your mutant HD5 is. So right here, here, and here. And I just wrote the word mutant above it, just so I don't have to think about what that diagonal box is, right? If I put the manuscript down and come back a few days later, I don't have to be confused. It's right there in front of me. And then we look at the X and Y axes. And even when you give a presentation, you always want to point your readers or your audience to the X and the Y axes and explain what they're seeing. So in this case, when we look at the X axis, I see this word, right? HD5, again, that stands for alpha defensin 5, and micromolar, that's a concentration. So automatically, I'm sort of assuming that these numbers are here. These are micromolar concentrations of adenovirus, of alpha defensin 5. So in my brief summary of the X-axis, I wrote, this is concentration of alpha defensin 5 increasing from left to right. And that's why we go from this 0 number up to this 50. And now I can make an assumption that the researchers, they use 50 micromolar amount of mutant HD5 in that one experimental condition. Also, everything here is color coded, but don't feel like you're restating the obvious. It's okay to restate the obvious. And I wrote down literally different ad serotypes, ad 5, ad 26, ad 35, and ad 48. So now we have a pretty good understanding of what's on the X-axis. Let's move to the Y-axis. And what it says is percent of control infection in EGFP plus A549 cells. So we already know what EGFP and A549 cells are because we asked those questions earlier. And we can feel confident about this. If you're giving a presentation and someone's like, well, why do they use A549 cells, right? People do that. You may be asked that. Well, now you have that in your biology repertoire and you can answer very elegantly because you do know, you're informed with that knowledge. So in addition to that, so the Y-axis is EGFP expression, and it says normalized to virus with no defense. And so I haven't really gone into that, but we have that as a line right over here. Experimental results are normalized to control infected with virus in the absence of peptide. So I'm sort of pointing that out right now, where I go to this virus here, right? This is ad5, and there's 0 concentration of defensin. So that's our control. It was normalized to 100%. So I look at this, I'm going to come right over to the Y-axis, and I see that 100% equivalent. The same thing could be said for ad 26 at the 0 concentration, right? So I'm stating the obvious for myself, and I'm just, bringing this figure to light. And you could probably do a lot more than this. And I have my take-home points up here. And I was very blunt with my take-home points. All I said was add 5 is inhibited, and that's with increasing concentrations of defensin, right? So as we go up in concentration, add 5 is becoming inhibited. And I used a traffic light, a red light, which means it's being stopped or inhibited, right? Just so, that makes sense to me. And if you were to present something like this to an audience, they appreciate that, right? You just, you want to make it as easy as possible for your audience to understand what you were describing. So add 35 had the same phenotype, and add 26 and add 48 were different, and that's why we put them in green. Now, there are other ways to understand concepts and manuscripts. Use YouTube. YouTube has a lot of really good video content out there. Now, these aren't peer-reviewed YouTubers, of course, but they do have a lot of good, relevant information. And another point, which is very powerful, I want you guys to discuss amongst your peers. And I can't tell you how powerful this technique could be. And let me just state it like this. So when I published this manuscript, right, the people who are on this list, these are my colleagues. I spoke with them on a regular basis about the work that I was conducting here, since I'm the first author on this particular manuscript. And we've had copious amount of conversations, right? This wasn't an individualized project. In a lot of ways, I had help, right? This guy, Alex, he was also a fellow postdoc at the time, who I'm very friendly with. Peter, he's a great friend of mine. He was actually a groomsman in my wedding, and I met him in the lab. Aaron and Makta, these are graduate students in the lab. Malika and Raphael, these are senior scientists in the lab who were helpful in a lot of ways. Zheng Feng, he was a very good friend of mine. Kevin, Sebastian, Sid, they were all master students who worked under my tutelage at the time. And then Dan Baruch, he's the head man behind the lab who had the funding. So the point is that I've had many conversations with my colleagues about this. So there's no reason why you can't do that with your peers. So my suggestions are, if you want to just set up a lit review discussion group, talk to the executive board members for this club. Talk to me. I could help you guys set these things up, right? Set up a meeting where you do this once a month, where you're with 5 of your friends, and each one of you are in charge of one scientific journal, right? I may have Nature, this person has Science, Cell, and you just report on the manuscripts or the data that you find interesting, that you think will be relevant to share with your friends. And you're going to start building your biology toolkit by doing that, because you can get practice and experience reading manuscripts. There's also other techniques that you could use. If you aren't familiar with this software platform called Perusal, it's fantastic. And there may be some alternatives to that, like Google Platform. You guys are much more tech savvy than the professors are. So you guys know much more about this than we do. You want to just find some sort of document or some sort of platform where you can put your document there, and then everyone can make comments on it. And here's an example of that. Using the Perusal platform, there are 4 individuals here who are making comments on this scientific manuscript. Whatever the scientific manuscript's about doesn't really make a difference. And this is the power of it, right? If you're in a group and you're making comments on the manuscript, right? Alan comes along and Alan's like, why is this the case? So whatever this is saying, why is this the case? I don't get it. And then David provides a very long, rational explanation to answer Alan. And then Alan could sort of move on in the manuscript now that he has a better understanding of it. And it's really a collective environment that you can create not only for yourself, but for others, and you could help each other. And so you don't have to feel like you're just in this alone when you read scientific literature. And I did just provide this brief little rubric to keep you guys focused and on track when you're going through scientific literature. And I challenge you to use something like this, or you make your own. You read the beginning of the introduction, and you just use enough words to fill this box. So you just provide a small summary. The middle of the introduction, you do the same. And then the last paragraph in the introduction, you do the same. So you're looking at maybe like 6, 7, 8 sentences among these 3 parts of the introduction. I'd even leave a box for the materials and methods. I think it's a waste of time unless you have a real reason to look at that. So don't even waste your time summarizing what the materials and methods are about. Results and figures, right? Maybe have 1 box per figure. So you have your 2-sentence or 3-sentence summary per figure. And you do the same thing for different parts of the discussion. So by the time you're done doing this, you're going to have a very succinct and concise overview of that manuscript. It might be 50 sentences or so. And it might be a little bit larger than the abstract. You could go back to the abstract at that point and then compare your interpretation of the data to what the abstract actually showed. So don't be, I don't want you to feel, discouraged when you look at the length and the complexity and the breadth of a scientific manuscript and literature. You shouldn't feel that way. Break it down into individual components. And I just want to show that there's some other resources available for you guys. So Lehigh University in our library services, they have articles over here on how to read scientific literature. And there's 5 other articles here. I've seen some of these in the past. It's a lot of really good information. I provided you with a different perspective, but learn as much as you can about how to break down scientific literature and then use that information, be more informed when you tackle this. We also have other resources through Lehigh's libraries on how to write a review article, how to interpret that. So there's information that you can take care with you. And with that said, so it looks like we're about 50 minutes or so. That's what I have. And that's literally just a brief snapshot on breaking down a couple components of scientific literature. As you can see, and why I stated earlier, I could spend 5 one-hour sessions really breaking things down. But for what this session was supposed to be about, I want to give you some snapshots and show you in the context of a real manuscript that some of you have actually read previously. So with that said, I want to stop talking. If anyone has any questions, please feel free to ask.